Hello everybody, thank you very much for joining us today. A big welcome to you all. I'm Nancy Holroyd from the Sanger Institute's Tree of Life programme, and I'm here with 5 great speakers and my co-chair Giulio Formenti, who is bioinformatics lead at Rockefeller's Vertebrate Genome Laboratory. Over the last few days, we've had some really good talks covering from sample to sequence and from sequence to assembly. And today we'll continue through the path of the genomics journey with 5 more talks, this time covering from assembly to genome. The talks will be played now, one after the other. Our first speaker will be Terence Murphy, talking about a new high performance and high sensitivity tool for contamination detection from NCBI. And then we'll hear from Joe Collins from the Sanger Institute, who will tell us about how complex genomic architecture creates challenges for manual curation. Next, we'll hear from Roderick Grigol from the Centre for Genomic Regulation in Barcelona with the talk Fast Pre-annotation of Protein Coding Genes. Then we'll hear from Surya Saha from the University of Arizona about workflows for rapid functional annotation of diverse arthropod genomes. And finally, we'll hear from Michael Markey from Wellcome Open Research, who's going to talk to us about the Tree of Life genome notes. Please do write your questions into the Slido chat during the talks, prefaced with the speaker's name, and we'll work through as many as we can after the talks. Enjoy the talks.
Hi, I'm Terence Murphy. I'm the team lead for the RefSeq project at the NCBI. Today, I'm going to tell you about some work we have underway on a new contamination screening tool. The sequence archives have seen really impressive growth in recent years, now exceeding 15 terabases of sequence and over 1 million assemblies from a wide variety of organisms. Sequence is doubling about every 18 months, so the archives will probably receive another 8 terabases just in the next year. When you submit to NCBI, we run a variety of checks to help ensure data integrity in the archives. We run some basic sanity checks, we validate annotation, can even add annotation for some organisms. We have a process to verify species for prokaryotes based on ANI, and we check for foreign sequence contaminants. This includes checking for adapters and common contaminants, and also looking for sequences from unintended organisms. These can come from environmental sources, like on the surface or in the gut, from food, intracellular organisms, or maybe the sequenced organism was infected, and also from lab contaminants and data handling errors. These contaminants can come from a wide variety of species, and failing to identify and remove them leads to bad data in the archives and errors in downstream data interpretation. Our screens find a lot of contamination, including nearly half of eukaryote genomes and 16% of bacteria. This includes both adapters and foreign sequences. So these screens do a lot of good, but we've long recognized they have a couple of problems. First, screening at the time of submission is really too late. You've already gone through many rounds of assembly and polishing. Maybe you've already done a bunch of work on the genome and written your paper. Now you submit, and we tell you, hang on, there's a problem. What you really need to do is screen early, and again when the assembly is finalized. And to do that, you need good tools that you can run yourself. Second, our current screens are designed for high specificity, and we know they miss some contaminants. How much? Well, it's hard to say for sure, but sometimes we find megabases of contamination during annotation, and we get user reports about problems. There have also been many publications looking at various types of contamination, with this recent study from Steiniger and Salzberg reporting over 2 million contaminant sequences in the archives. Now, half of this was from only 30 particularly bad assemblies. There is a little bit of noise in the results, but clearly there's a need for a higher sensitivity screen. So we have a big initiative underway at NCBI to improve our genomic resources. And one of the first projects we're tackling is to make a foreign contamination screening tool that you can run yourself. We're aiming for something that's easy and reliable, high sensitivity and specificity. and where you can run the same code that we use for screening submissions. This overall tool is still under development, but we're working to release parts early so you can start using them and we can get your feedback. The first two parts aren't quite public yet, but I expect they will be in the next couple of months. This includes an adapter screen based on our existing VecScreen tool and a new screen for foreign organism sequences. We've studied and learned from other approaches and data sets over the years, and we've come up with a new approach aiming to give us the sensitivity, specificity, and performance that we're looking for. At its core, it's based on a new cross-species genomic aligner that we call GX. It uses a modified K-mer hash to get matches on similar sequences. These seeds are then extended and resolved into full alignments. It runs with a database currently built from about 650 gigabases of genomic sequence from a wide variety of species, mostly using what's in RefSeq, but adding in more species where they're needed. The alignments are scored by both specific taxa and their BLAST divisions, aiming to classify sequences as either correct or contaminant. Now, rather than just looking at top hits, it analyzes data from multiple taxa to help resolve true versus false positives and help compensate for any contaminants that might be in the database. It does require a large RAM host. It takes about 15 minutes to load the necessary index files into memory on a GCP virtual machine. Once loaded, GX screens at 1 to 10 megabases per second, or about 8 minutes for a human-sized genome. That's about 3 orders of magnitude faster than our current BLAST-based screen, against 8-fold more sequence with higher sensitivity. So it's fast and seems promising, but does it work? Well, to validate, we've been looking at specific test cases with large data sets with various tools. Here's a bee genome with mostly a bacteria contaminant that we've looked at in great detail. And you can see GX finds nearly everything we can find with other tools or by deep manual review with no false positives. And looking across larger sets of assemblies, we see GX detects most contaminants found by other methods, missing around maybe 5 or 10%. It also finds many more, especially compared to our current production screen. These data sets include many of the most contaminated genomes around. So this is a very stringent test. So are all of these extra sequences really real contaminants? There is some noise that we're trying to reduce, but the vast majority of this is real signal. That's shown by a low rate of novel contaminants in our curated set. The extra contaminants that GX is finding tend to either be very convincing alignments by themselves, or we're finding more sequences from the same organisms that we know are contaminating, or they're organisms that make sense, like finding aphids in plants. So we think, well, specificity isn't quite perfect, it's very high, and we keep pushing to improve it. Well, our focus is on getting these tools out to the public to help with new assemblies. We started looking at cleaning up the existing genomes we have in RefSeq, first looking at the eukaryotes. Well, 80% have no or very little contamination. There are 43 with over a megabase, including one plant genome with 45 megabases of fungal contamination. We're reviewing the worst examples, either switching to better assemblies where we can, or reaching out to submitters to help revise some of these assemblies. We try to keep RefSeq assembly sequences the same as their GenBank counterparts, but in some cases, we may drop sequences only from RefSeq and have to leave them in the archive copy. And while we started with eukaryotes, we will be looking at prokaryotes in the future. So that's all I have time for. I want to thank a really great team of developers and subject matter experts, especially Alex Astatian, who devised and developed the GX contamination screen, the work of Steven Salzberg's group on Conterminator and other contamination analyses, Mark Blackster's group on Blob Toolkit, especially the NCBI genome submission team, and all the submitters to INSDC. If you're interested in being an alpha tester, please contact us at RefSeq Support. We'd love to work with you.
Hi, I'm Jo Collins. I work at the Sanger Institute as a curator in the Genome Reference Informatics Group as part of the Tree of Life project. Today, I'm going to present to you on how complex genomic architecture can create challenges for manual curation. Assemblies come into GRIP via the Tree of Life Assembly Group based at Sanger or via external collaborators. The TOL group takes the PacBio HiFi long read assemblies and processes them through their pipeline, which includes haplotype separation, polishing, and then scaffolding using Hi-C. Depending on how structurally complex a genome is, this will obviously have an effect on how easily it assembles. For some genomes, such as the Lepidoptera, passage through the assembly group pipeline typically results in telomere to telomere chromosomal units, with none or very few manual interventions required to achieve a fully assembled genome. Other assemblies come out of the pipeline in a much more fragmented state, such as the example here for the Blue-rayed limpet. All assemblies go through a decontamination process before manual curation begins. Then, using a number of tools, the available data is interrogated and errors are corrected, thereby providing added value to the end product. Ideally, following curation, a genome is fully assembled into chromosomes and matches its published karyotype. Upon completion of curation, assemblies are submitted to the INSDC archive for release into the public domain. As was previously alluded to, not all genomes are clear-cut. This is the Hi-C map for the Xanthogramma pedicequum assembly after it came through the assembly group. You can see that there are some large scaffolds, but when we look closely, we can see some typical assembly issues. For example, there are a number of misjoins that can be seen when we look at the off-diagonal signal. In addition, there are scaffolding errors, both where scaffolds have been incorrectly incorporated into the wrong chromosome, and also where sections of the chromosome have been joined in the wrong orientation. Although not ideal, the aforementioned issues are things that can more often than not be easily resolved via curation, with the information provided by the evidence data. A much larger issue in this assembly are the numerous small scaffolds, or shrapnel as it's often referred to. When we see this, we have to wonder why the automated process was unable to assemble this data into the primary scaffolds. Further scrutiny of the data reveals that compared to the larger scaffolds, the Hi-C signal for this shrapnel is exceptionally weak, which poses challenges for curation. So what might be the cause of the weak signal? Repeat density analysis for the assembly indicated that the majority of these small scaffolds have a very high repeat content. It is standard practice to filter out multi-mapping reads, as this can result in ambiguous Hi-C signal. Therefore, it's highly likely that the reads that map to these small scaffolds were discarded during the read alignment process, as they did not map uniquely in the assembly. So in order to include these reads in our assembly so that we don't lose any possible insights that they may give us, we can remake the Hi-C map, but this time include the multi-mapping reads. Of course, in dropping the filtering stage, we are likely to introduce ambiguity into our map, where we just have a very strong signal between the repetitive scaffolds, but we cannot decipher how they should be assembled. Even if the data is not granular enough to correctly assemble those repetitive scaffolds, it may be clear enough to at least localize them to a particular chromosome, which would be advantageous. This is a comparison of the Xanthogramma Hi-C maps, both without and with multi-mapping. What you can see immediately is that in the map using the multi-mapping reads, the shrapnel now has a much stronger Hi-C signal. As a result of this, it's now clear that much of this shrapnel groups together and is localized close to the ends of scaffolds 3 and 4. Even with this new information provided by the use of the multi-mapping reads, assembly of the data is not trivial. When we zoom in, we can see that many of the scaffolds are now exhibiting checkerboard patterning, indicative of TADS. This type of signal distribution makes curation of this data extremely challenging, as determining scaffold order and orientation is problematic without a clear Hi-C signal. The best strategy for this assembly was to try and group together the scaffolds based on the strength of their shared Hi-C signal. Fortunately, the pretext tool that we use to interrogate our Hi-C data has an editing function that allows us to move and reorientate scaffolds within the map. Once the scaffolds were grouped together, it was easier to see unambiguous joins, and also where these groups most likely joined onto the primary scaffolds. As the shrapnel was grouped, updated Hi-C maps were created, as larger scaffolds are easier to reposition within the map. Looking at the edited map, it became obvious from the off-diagonal signal that scaffolds 3 and 4 are actually part of the same chromosome. Following multiple rounds of joining, the chromosomal structure became clear. Scaffolds 3 and 4 make up the 2 chromosomal arms, joined by an extremely large heterochromatic region. With this chromosome assembled, the genome now fits the expected karyotype. So the strategy for this chromosome that was taken forwards to submission was first order the repetitive scaffolds in relation to each other as far as possible, then join the shrapnel as accurately as possible to the 2 chromosome arms using iterative rounds of curation, and finally submit a genome note with the created assembly detailing how the region had been put together, with the caveat that it's a region of low confidence. We've now used this best fit approach in order to assemble complex structures in a number of genomes. All were submitted with an accompanying genome note. In all of these examples, assembly of the marked region was only made possible as a result of using the multi-map read approach. As is evident, without this strategy, all of these chromosomes would have been submitted in a fragmented state. Unfortunately, the multi-mapping approach doesn't help resolve all complex regions. Some sequences are just too ambiguous to localize. One family where this seems to be a particular issue are the Hymenoptera, 2 examples of which you can see here, where after curation there remains a considerable quantity of unplaced heterochromatic sequence distributed over many small scaffolds. Only advancement of current technologies will facilitate the resolution of these types of issue. So to sum up, our approach, which utilizes the multi-map reads in the production of the Hi-C maps, means that we're able to visualize previously masked portions of the genome. Using this approach allows us to localize and to a greater degree assemble complex regions of the genome that it was not possible to discern when using the filtered Hi-C pipeline. Although we're not able to fully verify the assembly over these regions, use of a genome note accompanying the submission allows us to alert the user that there exists a region of low confidence in the assembly. Without the valuable insight provided by use of the multi-map reads, the end product achieved for a number of genomes containing these complex regions would, we feel, be less meaningful. Using this strategy means that in most cases, the expected karyotype can be achieved. This provides the end user with a final product that is simpler to navigate. There are exceptions to the rule. In some assemblies, the Hi-C signal remains too ambiguous to confidently place the heterochromatic sequence. I'd like to thank you for listening.
Good morning. I'd like to thank the organizers for inviting us to present our contribution as a talk. As you all know, the first step into translating the sequence of the genome to the biology of a species is identifying the genes and transcripts that are encoded into that sequence. That's not an easy task because of the complex structure of the transcripts in the genome, which essentially covering the entire genome, with alternative transcripts overlapping, nested in different strands of different types, etc. So the task is so difficult that, as you all know, 20 years after finalizing the human genome sequence, we do not have yet a final annotation of genes and transcripts into the human genome, despite the effort that consortiums like GenCode has been putting during all this time with thousands of researchers involved into the task. In general, what these methods use is a variety of sources of information that are combined into more or less sophisticated methods. I will not go over them, but the complex integration of these heterogeneous sources of information is underlying most of the more sophisticated methods for gene annotation, like, for example, ensemble. This said, it has become clear quite recently that while it is true that genes has many alternative transcripts, analysis of RNA-seq data across multiple conditions have revealed that in general, it is always the same isoform, which is the most highly expressed, the dominant one across all these biological conditions. Not only that when we look at proteomics data, what we actually see is that most human genes have a single main protein isoform, which is the same across biological conditions. So in a sense, what we can say is that a large fraction of the transcriptional output of cells corresponds to a constitutive single protein coding isoform per gene. This, of course, simplifies to some extent the task of gene finding if we pose it in a slightly different way. Of course, predicting one protein coding isoform per gene does not capture the full transcriptional complexity of eukaryotic cells. It does not capture relevant biology. It does not capture non-coding RNAs, it does capture alternative isoforms. It's a poor basis for functional genomics. Transcription sites are poorly predicted, and it's very impossible to really attach the correct promoters to the genes. On the other hand, it is true that it captures a large fraction of the biology of the cells, maybe 70, 80, 90% is actually carried by protein coding genes. Can be used for automatical functional annotation because the functions of proteins can be transferred from one to another, something that cannot happen with long non-coding RNAs. Computational methods exist with control accuracy, not necessarily with large accuracy, with accuracy that we know that it can be really employed directly on the genome sequence with any additional native data, RNA-seq or other functional data, and actually it can be performed very fast. So here I actually compare the predictions of 4 different what is called ab initio gene finders, so programs that predict protein coding genes, against I compare it against the annotations of ensemble 20 different invertebrate species across the entire phylogenetic spectrum and genome length. And as you can see, these genomes do not perform particularly well, right? They had okay sensitivity and specificity. Augustus is a program that performs at the top of these methods, but GeneD, which is a program that we developed, does not perform that either. But actually, the real advantage of this method is at the running time. So these methods can be used very fast. So GeneD in particular is very fast. It just analyzes genomes of the size of the human genome in about an hour, compared with Augustus, that takes 5 days. But I mean, if you compare this with the efforts like GenCode, that is for 20 years, right, trying to get the correct annotation of the human genome. So that's really a huge difference. And you could say, well, yeah, you can always parallelize this program, but it's true. But we also need to be aware that computation contributes to the carbon footprint. And I think that we need to be aware of these things, and bioinformatics applications need to be optimized as much as possible. So to run fast, not only because we want the results rapidly, because also we want to not to contribute too much to the carbon footprint. As I talk about GenCode, GenCode is an annotation of the human and mouse genomes, for which there is a lot of effort being devoted. And essentially because protein coding genes are particularly well conserved across genomes, can be leveraged to improve the annotation of other genomes that are other than the human and the mouse. And that's what we have done. Many of these ab initio programs allow to take external information as evidence. Essentially, in the case of GeneD, GeneD is just a program that efficiently change predicted exons, maintaining the frame consistency and maximizing some scoring function. And essentially what we do with GenCode, but we only do with GenCode, is we rescore the exons that overlap protein coding genes annotated in GenCode and boost their score. So this actually increases very much the accuracy of, well, at least the specificity of GeneD, that is the proportion of predictions that are correct, and with inaccuracy which is comparable or higher than Augustus, at a fraction of the computational cost. This is a more challenging evaluation. It's not 20 vertebrate genomes, it's 100 ensemble vertebrate species with assemblies that meet the AVP standards. There is room for improvement because these are the accuracies at the different taxa. And of course, GenCode is a mammal, and this leads to, in general, better predictions, although not necessarily, for mammalian genomes. And as we move away from mammals, the predictions are not as good. So by fine-tuning the parameters that we use to rescore the exons based on the overlap with GenCode protein coding genes, we can improve we believe that we can improve the predictions of GeneD. So we're working into a new version that does not require training, that can be employed, therefore, without training any eukaryotic genome, and can automatically fetch the most appropriate high-quality genome annotation for a reference species. So let me finish. Computational predictions of protein coding genes capture only partially the transcriptional complexity of genomes, but if minimally accurate, are sufficient for initial functional characterization of genomes. They serve as pre-annotation, on top of which more accurate annotations can be built. And in particular, if efficient enough, they can be deployed and run autonomously without the need of complex and centralized resources in any almost laptop. So let me finish acknowledging the people that have contributed to the work and the initiative. This is part of the Kata for the original project. which is an affiliated project to the ABP. Thanks for your attention.
Hi, my name is Surya Saha. I'm going to talk to you today about functional annotation for arthropod genomes. I'm going to describe a pipeline, how to run it, where you can run it, and how to interpret your results. First of all, thank you for coming to this talk. I hope you find this useful. So I want to spend a few slides telling you how to access this pipeline, because if you just take that away from this talk, that would be a good take-home lesson. So we have a Read the Docs site, which describes the pipeline in detail, all the steps, how you can run it on Cyverse, how you can run it using Docker or Singularity on the command line, and how you can, if you work for ARS, how you can run it on Series or Atlas HPC. As I already mentioned, it's on Docker Hub, so you should be able to download the latest release tag from Docker Hub. If you have any issues, a great way is to just contact us through the GitHub repo. This is all documented on the Read the Docs site. You can file issues here, or you can just contact us by email. You can email me. You can find my information in the conference materials. We have all this on GitHub, and we will, and the best way to file issues is to report it on GitHub or send us an email. You can find my contact information in the conference materials. And all the code we are using is open source, so that makes it much easier for collaborative development. All right, a little bit of background on this project. So this originated out of the i5K project. If you haven't heard about the i5K project, that is the Insect 5000 Genomes project, which is a collaborative community of scientists, government researchers, and sometimes industry from all over the world. And now it has grown into larger community with the Ag 100 Pest project, which is more of a USDA project. And so the goal here was that we have made a lot of progress, we as in a community, have made a lot of progress in sequencing and assembly of arthropod genomes. But the value of those genomes is only realized when you can assign function to those genes and experimentally validate those. So this is where functional annotation comes into the picture. And the gold standard is, of course, if you had manual curation of everything, but that is not possible for many, many practical reasons. So the goal of this project was to figure out a pipeline that works well for a diverse range of arthropods. That was the key. And then develop a pipeline which works in most use cases and provide it to the community. And we also have publication already. All right, so functional annotation tools. The steps are essentially in functional annotation. The structural annotation is step one, where the proteins are identified. Then we are assigning functions to them based upon sequence homology. So we have three different tools which are doing this. In step number two, it's Goanna. So the interesting thing, or the really valuable thing that Goanna does, is that it uses experimentally validated Go terms to assign Go terms to your genes. InterproScan is a nice complement where it uses motif search and gene family based annotation, which provides you broad level Go terms for most of your genes. And then in the next step, we have COBAS, which uses predominantly KEGG orthology to assign pathways. And we have pathways from Biopsych, Reactome, of course, KEGG. The information that we get for the Go terms from Goanna and InterproScan is combined in a GAF file, gene annotation format file, which is a commonly used standard for gene ontology annotation, which also includes evidence codes, which are very important to keep in mind. If you use a tool that assigns Go terms without the correct evidence code, then it is not great. And I can talk more about it, but we don't have time today. All right, so what do we get out of this? We get motif and domain information, which gives us an idea of the evolution of the gene families in your newly sequenced organism. These genes can be targets for manual curation. You can export the Go terms for doing Go enrichment for your experimental analyses. Then you have pathways, and you can use COAS or any other tool for doing pathway enrichment analyses. Measuring Go quality, I already mentioned breadth and depth. What I basically mean is that when you have Go terms, if they are at a very high level and broad, it doesn't tell you too much about the function of your gene. So you need deep Go terms to infer the correct function. And this is where InterProScan and GoANA are complementary. Because if you see this example where GoANA provides more than 8,000 Go terms for only about 700 genes, but those are mostly deep and high quality Go terms. And we measure that by using a GAF measure. So when you combine both InterProScan and GoANA, you get the best of both worlds. And that's what I'm showing you here in the third column. So to keep it within time, we are keeping this presentation really short, but as I've already mentioned, this has been published. You can find it in the latest issue online of Insects in the special genomics issue. And all the documentation is online on Read the Docs. So we are continuing to test it, and we are deploying the workflows on both Docker and Singularity on various platforms. Please implement it on your cluster or on your server and contact us if you have trouble. This also helps us, the function annotation can help users to identify missing or erroneously annotated gene families which need to be fixed. Training sessions will be provided at future conferences like ESA. We of course welcome feedback on tools and documentation. I would like to thank USDA for providing funding, and Fiona McCarthy at University of Arizona, and Monica Paulshaw at National Ag Labs, who is one of the co-leads of I5K. They were the leads of this project, and we are pretty excited to present this to the community, and we hope many people use it, and then so that you can do comparative analysis with your newly sequenced genomes. I hope this is useful. Again, the documentation is available on the Read the Docs site, and annotation for 15 diverse arthropod genomes is available at this NAL link. I'm sure the slides will be provided at some point, but please feel free to reach out if you have any questions about the slides or anything else about the pipeline. Thank you so much for your time.
My name is Michael Markey. I'm the Publishing Director of F1000, and I work very closely with the Wellcome Trust on their publishing platform, Wellcome Open Research. So what I want to talk about today is a project that Wellcome Open Research is working with the Tree of Life project on, and that is creating a new article type that is called a genome note, which we hope will help disseminate all of the information of the genome sequences from the Tree of Life project. So the Tree of Life on Wellcome Open Research, what we've been discussing is really a way in which we can use an open research platform, such as Wellcome Open Research, to share all of the genome sequences from the Tree of Life project in a public manner. So making them open access and ensuring that there is all the underlying open data and links to the raw sequences in the ENA. So what we've been discussing is this is possible and we would like to do it, but we also want to ensure that we can give credit to all the researchers that are involved, all the citizen scientists who have contributed to collecting some of the species, and also everyone involved in the sequencing itself. So we want to do that through a traditional publication route, so ensuring that all of the articles are as discoverable as possible, and that the researchers who are involved can get some credit for the work that they've done. However, due to the size of the project, so potentially the 70,000 species that will be sequenced, publish them in a very traditional way would be very expensive. So publication costs will be very high. And also there'd be a large burden on the community by traditionally peer reviewing such a large amount of content. So we've been working together to think about how we might be able to do this in a much more efficient and practical way. So what we've devised is a new article type called the genome note. So together, the F1000 team, the Wellcome Open Research team, and the Tree of Life team at the Sanger Institute have devised a streamlined approach where we're going to be making the publication much more automatic and hopefully doing it in such a way that we could at some point get to the point of publishing over 100 sequences a week. So how we intend to do this is really by automating a lot of the publication process. So we're going to create an XML pipeline that goes directly from the Sanger sequencers via an API directly into the Wellcome Open Research editorial management system. And so this automated pipeline will bypass any humans having to submit anything, and it will also enable us to do a much more less rigorous editorial check, as everything will be sent and all of the data that is required will be part of this XML pipeline. So once that XML from the Sanger sequences come into our editorial management system, we will then automatically convert that XML into HTML and a PDF, and we'll give the genome note and all the associated data sets a DOI. So everything that is needed for a traditional publication will be created. We'll also be hosting in-article interactive figures so that all of the users and readers of the genome notes can go into each of the article figures and they can then also interactively see how they work using the Blog Toolkit tools. Another big cost in publishing is peer review. So what we're also hoping to do is innovate here. So we're going to be having a mixture of what we're calling machine review alongside community review. And we're also going to make sure that all of these genome sequences are published in a genome note, which is then indexed in PubMed Central, which will make them much more discoverable and enable people to use the data and build upon those findings. So when I say peer review innovation, what we're hoping to do is that all of the information generated for the sequencers, so that's the BUSCO scores and the data that are in the Blog Toolkit figures, what we'll be able to do is take that information, lock it down and send it to the editorial management system, who will then pull out the summary data for the community reviewers, but also provide link to a zip file of the raw data of the sequences. This will help us to really verify the genome quality and the quality of the genome assembly. So what we would then do is ask a pool of community reviewers. So we're hoping to gather lots of experts across all different career stages and a really globally diverse pool of people to help us verify the genome assembly assessment. So I'll talk a little bit about that later, but what they will essentially be doing is assessing the genome assembly quality by looking at the figures and provided data and making sure that everyone else can access all of that data. So it's a really innovative way of trying to peer review something that doesn't necessarily need a traditional peer review. It's more of an assessment and verification of genome assembly. So we've already launched the project. So the Tree of Life Gateway is now live on Wellcome Open Research, and it launched at the end of May. The links to the website is on the slides. We've already published the genome sequences of the Eurasian river otter, the red squirrel, amongst others. And these have been uploaded or submitted manually to start off with, because we were just looking at the format. But what we're hoping to do is that exact format will be part of the XML pipeline I just discussed. And all of the genome notes in this gateway will be supporting the Darwin Tree of Life project, the 25 genomes, the 25 years project, and the Aquatic Symbiosis Genomes project. So just to talk a little bit more about the community review, we really want to get the whole global genomic community involved. And what we're asking, as I mentioned, is to assess the machine review, the BUSCO and the Blog Toolkit plots, and verify access to the data. And the benefits of this is that all the reviews will be publicly available, so the credit can be due to the reviewers. They'll be linked to ORCID IDs. There'll be acknowledgement and to the contribution to the project, and it'll also be a chance for PhD students and early career researchers to hone their reviewing skills. So we'll be sending out a registration form and building our community pool very, very shortly. So you'll see some communications around that in the next couple of weeks. But if you are interested in being involved now, then please do get in contact with us at Wellcome Open Research. And we really are hoping to build a real global community to help us review all of these genome notes. So it's exciting stuff to come, and looking forward to talking more about this project as we develop it going forward. Thanks for listening.
Welcome back, everybody. Thank you very much to all the speakers for their really excellent talks. We have 10 minutes now for some questions, and there's a few that have come in during the talks into Slido, so we'll start with those. If we start with a question for Joe from Mark Palmeida Flores, who thanks you very much for the explanation and the tips on how to use the Hi-C data. He'd like to know when you can say an assembly is finally at chromosome level, and how you evaluate when to stop curating manually. And that's something I'm quite interested to know as well.
Okay, so we're very fortunate. We get very good assemblies come through our pipeline.
Go quality, basically mean level and broad.
We have some noise interference there. So I think it's gone now. Sorry, do try again,
That's
Joe.
okay. I was a bit confused. Yeah, so basically we're very fortunate. We have very good assemblies come through our pipeline. So when we're thinking about when it's at a chromosome level, we're looking mainly at our Hi-C data to see how the scaffolds are forming. And often we have fortunately enough, we have a karyotype to go with our data, and what we're mainly looking to see is when the scaffolds that we've got, depending on how good the assembly is, if you're able to curate those scaffolds and make all the joins and the breaks and things to create sort of single units, then that's when you're looking at your sort of chromosome level assembly. Fortunately, with things like the Lepidoptera, often, as I mentioned in the talk, we have telomere to telomere sort of scaffolds. We're fortunate in that we have additional data that we can lay onto our Hi-C maps to give us this kind of information. And in terms of when we stop curation, that's basically we look. I mean, it's generally Hi-C is probably the most powerful tool that we use for curation. And as I mentioned, we use both HiGLAS for Hi-C, and which is very good for detail, looking in very close detail. Gives us a very high level of granularity, but we're also very fortunate that we use the pretext tool, which enables us to sort of edit the map so that we can move things around, which makes our lives a lot easier when we're dealing with very fragmented assemblies. So the time that you stop curation and that you sort of call it, and that you consider that you have chromosome level assembly is when you have made all the joins that you can make in your Hi-C with your Hi-C data. Sometimes, unfortunately, there will be scaffolds that you are unable to localize. Sometimes you can localize them to a chromosome, but you cannot put them into the chromosomes in terms of mapping. And so we have to just leave some shrapnel if it's unplaceable.
Okay, thank you very much, Joe. A question here for Roderick from Peter. What data do you need to train a model for a non-model organism for gene ID? For example, Breaker recommends transcript intron locations. Is there an optimum input data set?
Well, the optimum input set would be long-read RNA-seq from as many different body locations as possible from the model organism, right? If this is not possible, then it could be long-read RNA-seq from the whole body, as deep as possible. And if this is not possible, short-read RNA-seq data, again, from the different organs or from the whole body. Additionally, of course, you could use genome conservation with species that are close enough, which are referenced and have been well annotated. And what we are aiming, if you just want to do if you are actually interested in protein coding prediction, what we are aiming is to have a method that actually doesn't need training. So essentially, the way in which you detect protein coding regions in genomes is because they show a particular bias in the composition of the sequence, the codon composition, right? It's different coding than non-coding regions. And this depends, this is specific of each species, but we have some ideas on how can this be generalized, because this depends mostly on the CC content of the genome. And I think that this could help into have training, into have ab initio predictors that do not require extensive training, which is one of the difficult things.
Yeah, absolutely. Thank you. We have a question here for Surya from Camille Jaron, who asked who mentions that you've advertised the annotation workflow for all arthropods and asked whether you've explored how well it performed on non-insect lineages. For example, Camille's springtail genomes are in a desperate need of a better gene annotation. Function is unknown in practically all of the genes.
So, right, I mean, looking under the lamppost is always easiest. So for the arthropod genomes, I can say we essentially tested out a bunch of, this is hopefully helpful, but let's see. So we tested out the different reference genomes we have for arthropods. So we looked at honeybee, we looked at tribolium, we looked at Drosophila, and we settled on Drosophila. So before choosing which reference you want to use for your experimentally validated Go terms, you would want to do a few tests. So I don't know what springtails are related to, but whatever you typically use as the protein reference, if you were to go and do some tests with your protein set, judge by the level of similarity, and then decide a reference set. Now, for Goanna, we have a database of experimentally curated Go terms. And if you have, you can look that up in the documentation. If it's confusing, please send me an email and I'll point you to the list of species that have experimentally characterized Go terms. And hopefully one of those will be relevant enough for you to use.
Thank you, that does sound very useful. A question I've got for Michael, do you anticipate that Genome Notes would catch on outside of the Tree of Life and that the approach that it's taken?
So that's a good question. I think the approach it's taking is bespoke in the way that the data is coming from the Sanger sequences, but the kind of pipeline that we're building should also be usable for other sequences too. So the idea is that, yeah, we're just trying to get that kind of information and stopping the kind of obstacles of having to upload things and making sure that the data's there. And by having that in a quick package that can then be sent and published within a few days is ideal in this situation. So what we're trying to do is refine it at the moment, and then hopefully we're going to make that pipeline available to others to use as well. So yeah, hoping that it does have a use outside of the Tree of Life project.
That's encouraging, I guess, considering the exponential growth in genome projects that we're anticipating seeing over the coming years. So that's good to know. A question that we have in Slido for Terence from Manny Mose: how does the FCS pipeline deal with HGT, so horizontal gene transfer events, and false positive contamination?
Yeah, that's definitely something that we've been looking at very closely. So our new screen with the GX aligner can detect chimeric sequences, so sequences with no gaps that look like they are part correct and part contaminants, and you can divide those further into sequences where the asserted contaminant is internal. So it has good sequence on both sides and it's something in the middle. That particular characteristic would be where I would start looking for possibilities of horizontal gene transfer. And it's rare. I have found some real cases. It's something that we'll probably look at mostly as a reporting presentation issue, saying maybe this is something wrong, maybe it's horizontal gene transfer, and maybe you should go look at it a bit more. But really, to resolve that, you need to know more about like what the raw reads were doing, and it's also really useful if you have neighboring assemblies to see if it shows up in multiple assemblies. I found a case just recently in a Drosophila assembly that was a small, like 1 kb bacteria HGG case that I could verify through other assemblies.
Okay, thank you. We will leave it there to stay on time. I'm sorry that there are still some questions in there, but I encourage the speakers to check those out and reach out to people who have some questions unanswered. I see that, Terence, you've put a contact in there for NCBI contamination testing, so please check that out if you're interested in that. So it's now time for a coffee break, and I encourage you all to join the spatial chat available on the networking tab on the portal. Thank you again to our speakers for some really great talks, and thank you for joining us.